Hello, this video is a guided tour of the DNA Subway Blue Line. The overall goal of the Blue Line is to help you clean up, analyze, and taxonomically identify the DNA sequences from samples you've collected. The DNA Subway Blue Line has three main stops. Select, where you'll upload and view the sequences you want to analyze. Edit, where you'll clean up and assemble your sequences. And Analyze, where you'll explore your sequences using some basic bioinformatic tools like BLAST, Muscle, and fill up phylogenetic trees. Please follow the directions in this video to analyze the practice set of fungal DNA sequences posted in this week's module. Do your best to follow along in DNA Subway and pause the video to answer the questions that are embedded in the video. This will help prepare you for your in-class analyses of your group's data coming up soon. Just as a quick recap of where we've been, at this point you've collected your specimen. You've extracted DNA from the tissue from that specimen. You've collected metadata and photographs. You've PCR amplified the data, sent it off for sequencing, and now that you have those sequences, we can begin to analyze your sequence data using the Blue Line of DNA Subway. First, you'll need to go to DNA Subway website using the links on Canvas or just by Googling DNA Subway. Once you create an account and log in, you can hit the Create a New Project button located in the upper right hand corner. This will take you to a screen that looks like this, where you can type in a project title, try to put in something informative, and a more detailed description that will help you refer back later if you create more than one project. Make sure once you add the project title and description, you select the correct sequencing type from the toggles, that's Sanger sequencing, and then click the blue Create Project button. You should see a screen like this. This is your DNA Subway project. The first stop is Select, where you'll upload or want to upload samples or files from your computer. We'll be using a practice set of fungal DNA sequences posted in this week's Canvas module using the link that says Practice DNA Sequence Files. If you follow that link, it should open up a Google folder that looks like this, where you can select all of the sequence files by holding down Command or the Shift button to select all of the files, and then right-click to download the files. It'll zip all of them together if you're on a Mac, sometimes on a PC too. Then you can open up that zipped file to wherever it downloaded to, and you should see 4 AB1 files. At this point, you don't have to open those files, you just need to know where they're located. So figure out where they are on your computer so that you can find them when you need to upload them onto DNA Subway. Once you figure out that pathway, you can go back and hit Upload Files to your device, follow the appropriate pathway to find those files that you just downloaded, and select all of them using the Command or the Shift button. Make sure all 8 files are collected and hit the Open button, and then they should be uploaded to your project. As you can see here, all 8 samples under the Current Files tab. So we'll be using a practice set of fungal DNA sequences for this video's activity, but of course we'll use the same method to upload all of your group samples when you do this in lab in the coming weeks. Make sure that you download all the forward and reverse sequences for each of your samples. So if you have 4 samples in your group, it'll be 8 total files. All right. Here's the first quick review question. Why does each person's sample have 2 files or reads? For example, Jessica has a forward read and a reverse read, as does Louisa, Mike, and Trey. Please hit pause until you have the answer. All right, so there's 2 files for each sample because they represent a forward and reverse read of the region of double-stranded DNA we're interested in sequencing. Having sequences for both the top forward strand and the bottom complementary reverse strand helps to ensure we have accurate reads for the entire region we want to barcode. Essentially, the 2 reads double check that the region where the forward and reverse sequences overlap are complementary to one another and therefore those individual nucleotides that match up are correct. So now let's talk about how to view the quality of each of your individual reads. First, let's talk about what high quality sequence looks like. Here you can see 4 electropherograms stacked in order of quality from high down to low. Each little wave in these images represents the fluorescent signal from an individual ddNTP nucleotide as it runs through the gel in a Sanger sequencer. The color of each wave represents a different nucleotide, A, T, G, or C, which ultimately gets translated into your sequence. Hopefully it's clear that the waves are clean and clear in the high quality sequence at the top and become increasingly ugly and garbled and uninterpretable as you move down to the horrible sequence at the bottom. From there you can go to the upload and drop down and you can see all of the samples that you imported. Each of these sample names represents a sequence, and you can see some of them might have a little warning with an exclamation point. We'll talk about that in a bit. If you do the toggle, you can click View Sequence, and then you can view your sequence. Typically, when you look at a sequence, you're almost always on the front end going to have a bunch of ends, which represent bad calls or bad sequence, and you'll see the same thing at the back end of your sequence, a bunch of ends at the end. But typically, all the stuff in between those two front and back tails look good. If you click on AB1 view, you can see the normal sequence view or the electropherogram. Running along the top will be the individual base calls. In the middle, you'll see a quality score for each of those base calls. The blue bars represent the quality for each individual call, and the small blue line running down the middle represents a threshold, what's called Phred score. You can also see the position of each of those base calls or account for each of those base calls. And you can scroll across and you can see that at the far ends, there's typically a bunch of ends with very low bars or Phred scores, not surpassing the threshold for high quality. Whereas in the middle, you see most of those calls have clean and high Phred scores. Now let's go down and take a look at Louisa's reverse read that has that warning sign that indicates it's a lower quality sequence. Here you can see, similar as before, there's a bunch of ends at the front end and back end, but there's also a lot of ends that we're seeing in the middle of the sequence. This is typically indicative of a lower quality read. Let's take a look at the AB1 view, and what you'll see is the waves at the sequence at the bottom are all garbled, overlapping each other and not clean. And the individual purple bars or Phred scores for each of those calls rarely pass that Phred score of 20 threshold. And so most of these calls are low quality calls that can't necessarily be trusted. Question: how many of the 8 practice reads are low quality? Please hit pause until you know the answer. So the answer is 1. Only Louisa's reverse read. That's the only practice read that has one of those exclamation marks. Next question: what exactly does it mean if a read has the low quality exclamation mark next to it? Please hit pause until you know the answer. A low quality score alert means that the average error rate for this particular sequence is greater than 1%. This indicates that the sequence is of low quality and may produce erroneous analysis results. So in other words, the sequence is less than 99% accurate. Technically, it's still okay to analyze these sequences further downstream if the software enables you to, but just take those interpretations and analyses with a grain of salt because they may be producing and are likely producing erroneous results. A quick review of Phred scores or Phred quality scores. Recall that these are the probability of an individual base call in a DNA sequence being correct. The Phred scores are indicated here by these lines and bars. Now the blue line represents a Phred score of 20. And if an individual bar exceeds that Phred score of 20, it means that that individual base call has a probability of greater than 99% of being correct. And typically a Phred score of 20 is the threshold for an individual base call to be considered of good quality or 99% accurate. So once again, if gray or purple bars in your electropherogram of your sequence pass above this blue line, it means they have a high enough Phred score or are accurate enough to be trusted. So given the information from the previous slide, which base call in the image above is most accurate? Base call A, B, or C? Please hit pause until you know the answer. If you said C, you're correct. C is the most accurate. It has a Phred quality score above 20 and therefore has a greater than 99% probability of being correct. Both A and B have Phred scores just below that threshold. Next question. How long is Jessica's reverse read? Please hit pause until you know the answer. Hopefully, you were able to figure out that you could toggle to the end of her read using this little bar here on the left and scrolling all the way over to the right. And you should have seen that the read ends at about 670 base pairs, as you see in the red arrow on the right. Now that we've uploaded our sequences and taken a general look at their quality, we'll go to the next major DNA subway stop, which is the edit sequences stop. Here we'll clean up and assemble our sequences for downstream analyses. So this will involve trimming the sequences and pairing our forward and reverse sequences to build what's called a consensus sequence. So the first stop here is in what's called the sequence trimmer. As we described before, typically in any individual read, there's going to be bad or no calls at the front end and the back end of the sequence, as you can see highlighted in the ends at the 5' and 3' end of this sequence here in the red boxes. Those ends typically need to be trimmed off, and Sequence Trimmer does that for you. It basically snips off these bad reads automatically for you. Let's take a look at Sequence Trimmer in action. Before we actually do the trimming, I just want to show you a couple of the sequences and what they look like before they've been trimmed, and remind you, here's Trey's reverse read, and it's got a bunch of ends on the front and back end of the sequence that are going to need to be trimmed off. Let's look at his forward read, same deal, a bunch of ends on the 5' end and a bunch of ends on the 3' end. Okay, so remember what those looked like before. Now let's go to Edit, Trim Sequences, and all you got to do is hit the magic blue button that says Auto Trim All, and it will trim all of your sequences for you. So now it's trimming off those ends on the front and back end of each sequence. And if we go back and look at the sequences, we're going to just look at Trey's. Now those ends are gone off the front and back end. For the reverse read, same deal for the forward read. And it'd be the same if you looked at any of the other sequences. They've all had the ends trimmed off. Just to look at this one more time, you can see a before and after comparison of Trey's reverse read. In the before, in the red boxes, you can see the end tails on the 5' and 3' end. And after trimming, you can see that those ends are now gone. Now that you've trimmed your sequences, answer these 3 questions regarding Jessica's forward read. Hit pause until you know the answers. All right, so hopefully you figured out that Jessica's trimmed read is now about 630 base pairs long, and it is of course shorter by about 30 or 40 base pairs than it was before, because some of those no calls or ends have been trimmed off the front and back end. And of course the quality should be higher now that those ends are gone. Now that our sequences are trimmed, we can begin to pair them together using the pair builder. So pair builder, the goal here is to link and align your forward and reverse reads for each individual sample. So right now, the way the software sees your samples or your individual reads is the forward read and the reverse read are sort of independent reads that it just knows what the 5' and 3' ends are of each of those reads. So essentially you have to tell the software which read is the forward read and which read is the reverse read, so that it can flip them over and start to align the sequences and make them complementarily match to one another. So you're essentially telling the software, this one's my reverse read, flip it over and try to align it to match it up with the forward read for the same sample. Now what Pair Builder also does is it will change the reverse sequences to the complementary bases, essentially making a single forward consensus read ultimately. So you can imagine the reverse read at the bottom right corner of this slide looks like that. What the software will do is switch each of those reverse reads to its complementary base pair and then align them together like you see in the forward read at the sort of top right of those sequence of images so that they all sort of match up. And then it will meld that into one individual sequence called a consensus sequence. So let's go ahead and pair our sequences up. Under Edit, you'll hit Pair Sequences. It'll be tempting to hit Auto Pair All, but that won't work. Instead, what you're going to do is figure out which is the forward and reverse read for each sequence. Mark the reverse read by clicking over the R until it turns red. Then select the little toggles or the check marks to the left to pair up the two that you want to pair, and then give them a label. So here for Mike, we're clicking the reverse read, checking those two boxes, putting in Mike as the sample name, and then hitting Save Pair. And you can see that each of these is now getting paired up for the next step. The next step is to edit what's called our consensus sequence. And a consensus sequence is essentially a single sequence made from the agreement between the trimmed forward and trimmed reversed read. So if you look here, this is a good representation of what our consensus should be. It says this is the set of sequences and individual nucleotides for our forward read, and this is those same nucleotides according to the complementary bases of our reverse read when we align the forward and reverse. And essentially what your consensus sequence does is look at the agreement between those two things to assess the overall accuracy of your final sequence. Before we use the consensus editor in DNA Subway, I just want to orient you to what it's going to look like. This will be the output in DNA Subway. The top strand here in Consensus Editor is your forward read. The next strand down is the reverse read, and they've been aligned. And then the bottom sequence with the colored letters is your consensus sequence. A few important things to note in the Consensus Editor screen. Lower quality base calls are indicated by lowercase letters, like the lowercase g pointed to by the red arrow here. And recall that a lower quality base call is one with a Phred score less than 20. In other words, it is less than 99% accurate. You also might see highlighted regions. These indicate mismatches, or places where the forward and reverse reads don't match one another. For example, you can see those two highlighted G's are G's in the forward read, but A's in the reverse read. Typically when this happens, the software automatically selects the nucleotide with the highest confidence in that position, or the highest Phred score in that position. And if you were to click on any of those individual letters in the consensus sequence, you can actually look at the electropherograms and compare the quality of the calls at those positions to see which letter was selected. So I'm going to do a very quick demo of how to trim and edit your consensus sequences. But before we do that, I want to remind folks that for the purposes of this course, we typically recommend that you only trim tails of your consensus sequence if greater than 400 base pairs remain after trimming them. If not, you probably shouldn't trim, or at least ask your instructor. So let's take a look at what I mean by that and how to edit your consensus sequences in this video. So here you see Jessica's sequence. You can click on a letter and it will left trim. You can click on a letter in the consensus sequence at the other end and it will right trim. And you can see that there's still 588 base pairs remaining. So let's look at Mike's or Luisa's sequence. Once again, what we're doing here is we're trimming off the regions where there's no overlap, because we can't necessarily trust the accuracy of the sequences in those regions because they're not being double checked by both a forward and reverse pair. So where there's not overlap, we're trimming off those ends, and if it's
remaining greater than 400 base pairs in length, we're keeping the consensus sequence. So here, let's look at Mike's. We're trimming off that tail and this tail where there's just a single read. And when we're done trimming, the trim consensus length is only 194 base pairs long. So I'm clearing those trims and just saving the untrimmed sequence for further downstream analyses. Whereas if we go to Trey's sample, here we can trip off that end, we can trim off this tail end where there's no overlap in the consensus sequence, and at the end, the trim consensus length is still 663 base pairs long, so it's okay to save that trim. So I've done that for each of these sequences. Once you're done, you can go back to Select, and you've now got consensus sequences under your current files, and you can view and copy those individual consensus sequences by clicking on View Sequence, and then Copy to Clipboard. So if you need to paste this into your lab notebook, your final consensus sequence, you can copy it to your clipboard and paste it into your lab notebook that way. As a quick recap of the edit sequences stop and sequence assembly with the goal of making the longest, most accurate sequence, there's been 4 main steps. Number 1, you've taken a general look at your DNA sequences to see their sort of quality. Then you've trimmed the sequences to get rid of the bad calls on either end. Then you've paired up the sequences, and then finally built and trimmed a consensus sequence to have your most accurate DNA sequence for your downstream analyses. So the last major stop in the DNA Subway Blue Line will be analyzing your sequences. First, you'll use a tool called BLASTN to find DNA sequences that closely match your barcode. It's sort of like a Google search for DNA sequences. Once you find those closely matching DNA sequences, you'll compare them using 2 different analyses. One called a muscle alignment that'll align all the sequences and see how they compare, and the other be building phylogenetic trees to compare your sequences and look at their evolutionary relationships to one another. The first step will be blasting your DNA sequences. BLAST stands for Nucleotide Basic Local Alignment Search Tool. What BLAST does is it searches for your consensus sequence against a national database of known sequences called GenBank. It's essentially a Google search for DNA sequences. To run BLAST, it's pretty simple. Just click each sample you want to blast from the drop-down in the BLAST tool, and then hit the BLAST button. Once you hit that button, it'll take a few seconds, and eventually you'll see a screen that pops up that looks like this. Then all you have to do is hit this little square. Once you hit that square, you'll have the BLAST results pop up that looks something like this. So this is what your BLAST results will look like. Here's the BLAST results for Jessica's unknown. For each sample, you'll have a list of the database hits ranked by what's called bit score. The bit score is calculated based on the alignment length and the mismatches, which you can see listed on the right side of the table. Alignment length is just the length of the sequence used to make the match, and mismatches is just the number of mismatch bases between the query or Jessica's DNA sequence or consensus sequence and each of the hits. So for example, that first hit, Pluteus ongoi, with a bit score of 1061, had 588 base pairs in its alignment, so 588 base pairs matched between that hit and Jessica's consensus sequence, and there were 0 mismatches. You can also see this E value or expected value. This is the likelihood that the match or the hit that BLAST made between our query and that sequence happened by chance. So the lower this value is, the better it is. And typically these values are way, way, way below 0, which means it's not by chance that the hit matched your consensus sequence. A few quick rules of thumb regarding BLAST hits. Number 1, if the number of mismatches divided by the alignment length is greater than 1%, this often means that the match is a different species. Though this is really highly variable across taxonomic groups and hotly debated. This is just a sort of loose rule of thumb to use for this course. Also, if your alignment length is less than 400 base pairs, your data often are not reliable, at least to ID to the species level. Though once again, this is highly variable depending on the primers you use, the region that you're amplifying, the taxonomic groups that you're comparing, and also hotly debated. This is just, once again, a general rule of thumb to use when you're IDing your sample for this course. If you click on an individual hit's species name or the link for a species hit, like here, Pluteus ongui, you can see some photos, some other taxonomic information that'll pop up. From here, if you want to do a little bit more research on the individual hits, you can highlight the species name and then just right click on it and search Google for the genus and species name. And oftentimes you can get good taxonomic information from iNaturalist or Wikipedia to help flesh out the information that you need to fill in your lab notebook. I've found that asking ChatGPT can also sometimes be useful to get this taxonomic information. So what do you do with all of these hits? Eventually what you're going to need to do is select particular hits to add to your project for downstream analyses. So for example, looking at the BLAST hits from Jessica's Unknown, typically what you're going to want to do is add the top 2 to 3 hits that are ID'd to the genus and species level. And this can be true even if it's the same genus and species for those top 2 to 3 hits. So for example, for Jessica, I'm going to select these top 2 Pluteus ongui hits. They have the same bit score and they are ID'd to the genus Pluteus and species ongui level. In addition, you want to add the remaining 2 to 3 taxonomically unique hits as rated by bit score. So top 2 to 3 is rated by bit score that have a different genus or species name. So for example, this is Pluteus petacetus, so it's a different species in the same genus, so I'll select that one. Another important detail to take note of when you're selecting hits to add to your project is you don't want to add samples that aren't ID'd to the genus or species level. So I wouldn't add this hit that says Pluteus spa or the hit below it that says uncultured fungus, because those are not going to be useful to help ID your specimen. So let's see what hits we would select for Trey's sample. So I'm going to go ahead and select Trey's BLAST hits and take a look at those. And here you'll see that the first couple of hits as ranked by bit score include the genus name Hymenopellis, but only SPA, so they're not ID'd to the species level. So I'm going to ignore those top 3 hits because they're only ID'd to the genus, not species level. And I'm going to add the top 2 hits that are ID'd to the species level instead. That's the 2 that are selected with a bit score of 1086, Hymenopellis liminospora. Then I'm going to add the next 2 to 3 hits that are ID'd to the genus level and taxonomically unique, which is Hymenopellis megalospora, and is likely found later on the next page and not on the first table that you see. So now that you've seen those examples, why don't you go ahead and try selecting the BLAST hits for Louisa and Mike on your own. Once you've selected sequences to add to your project from your BLAST search, the next step is what's called a muscle alignment. Muscle stands for multiple sequence comparison by log expectation. Essentially what a muscle alignment does is it takes all of the sequences that you've selected to add to your project and it aligns them or stacks them up on top of each other to compare them, and from there you can impute potential evolutionary relationships and relatedness between the sequences. So the first step in creating a muscle alignment is to add reference data that taxonomically match your samples and primers, but are not necessarily close BLAST hits. This allows you to select downstream what's called an outgroup, and I'll explain that to you in a little bit more detail very shortly. So to add reference data, what you're going to do is click the Add Reference Data, and then you'll have several choices of reference data to choose from. COI for animals, RBCL for plants, 16S for bacteria, and then ITS for fungi. So since we're analyzing fungi right now, we're going to select reference fungi from the ITS group. Once you've added your reference data, then you can select sequences to align. And this will essentially be all the blast hits that you selected from the previous activity, your samples in an individual outgroup. So this is how you'll do that. So this is going through right now, selecting all of your blast hits that you selected from the previous activity to add to the project. Then your consensus sequences of your unknown samples that you collected, and then an individual outgroup. And in this case, we're just going to select rust fungus, or sorry, smut fungus, and then hit the muscle button. And it'll take a few minutes to analyze. After about a minute, you'll get an output that looks like this. You're going to want to trim the ends off the alignment. So go ahead and click the auto trim button, and you should get a trimmed muscle alignment that looks similar to this. Here's your trimmed muscle alignment. Each row in this muscle alignment represents the DNA sequence of your samples or an individual hit from your BLAST search. The labels to the left of the alignment identify each sample. The numbers above the alignment at intervals of about 100 represent the base pair position in the sequence. The light gray areas in each column represent base pairs that match in each sequence. The colored areas represent polymorphisms, or locations where the base pairs are different across sequences. Each color is representative of a specific nucleotide. A's are green, T's are red, C's are blues, G's are black. The top row that says sequence conservation represents how often there are differences in the nucleotides at that position across all the samples you're comparing. The more white you see, the more often we see a nucleotide that is different from the most common nucleotide at that position. The second row down that says sequence variation shows you all the different nucleotides that are present at that position in the sequence across all the samples that are being compared. The third row down, labeled consensus, shows you the most common nucleotide at each position in the sequence. For instance, if you align 5 sequences and the nucleotides at position 20 are A, T, A, and G, then the consensus sequence should have an A at position 20 because A is the most common nucleotide at that position. All sequences are unknown, reference, and close matches are being compared to each other. The consensus row doesn't really need to be understood in detail, but it represents the nucleotide that matches in all sequences where a polymorphism or colored line is not shown. Lastly, dark gray areas, if you see them in your alignment, represent gaps or spots where the sequence is missing from a particular sequence in the alignment. These gaps can be created by the insertion or deletion of large chunks of DNA in some but not all of the species being compared. If you hit the ATCG blue button in the upper right hand corner, it'll zoom in on the muscle alignment so you can actually see the individual nucleotides labeled by the letter. So go ahead and click that in your DNA Subway screen and then answer these questions. Go ahead and hit pause until you've got the answers. Okay, so what is the maximum number of polymorphisms possible at any one site in the barcode? Well, the maximum possible number is, of course, 4. This is the maximum number of individual nucleotides that can be at any one site. It can either be A, T, G, or C. What is the most common base across all the sequences at site 5 is indicated by the arrow in the upper left corner there, site 5. The answer is A, as indicated by the consensus sequence. A is the most common letter if you compare what letter is at that point across all of the sequences being compared. And what does tray, or sample number 4, have at site 5? The answer indicated by what's in that blue square is once again A. And that's because Trey doesn't show a letter there because he matches the consensus nucleotide there, which once again is A. If you don't see a letter there, that means Trey, or whatever the sample is, has the most common nucleotide, or the same as what's listed in the consensus sequence. You're likely asking yourself, okay, so what do I actually need to pull out of this muscle alignment? The two big questions that you'll need to answer from this muscle alignment are, number two, which of all the taxa that are lined up is most similar to your particular unknowns? So in this example, let's say, which of these is most similar to Trey's unknown? And you'll also need to identify which taxon or which individual sort of sequence line is least similar to all of the unknowns. So if we look at Trey's sample, it's tough to say which one is the most similar, whether it's Hymenopellis incognita, Limnospora, Megalospora, radicata, or orientalis. Likewise, it's very difficult to say which taxon is the least similar across all of our unknowns. So a really useful tool to help tease this apart in a quantifiable way is the sequence similarity table. So let's look at that on the next slide. So once you click on the sequence similarity table, you should see something like this. This is a % sequence similarity matrix. It's a table that does a cross comparison of every single sequence that you're looking at in your muscle alignment. Sequences labeled 1 through 24. So for example, we know that Trey's sample is sample number 4. And so we can look at the highlighted area here and follow that row the whole way across to see which % sequence similarity is the highest between Trey's sample and the samples that we're comparing him to. And what you can see is 96.77 is the highest number. So samples number 5 and 6 are 96.77% similar to Trey's sample. They have the highest % sequence similarity in that muscle alignment. And so those samples, Hymenopellis, are both Hymenopellis limnospora. And so they have the highest sequence similarity to Trey's sample. We can also begin to decipher which taxon is the least similar to all of our unknowns. Can you hit pause and figure it out on your own? If you said sample 1, you're correct. If you look at the % similarity of sample 1 to all the other samples, you'll find that sample 1, the SMUT, is the least similar, about 55 to 67% similar across all our groups. So sample 1 will be our out group. And apologies that it says Baker's yeast there, it should say SMUT. SMUT will be the out group. Here's the sequence similarity matrix for Mike's sample. Which taxon is most similar to Mike's unknown? Go ahead and hit pause until you know the answer. All right, so if you said sample 20 and 23, which are Pluteus cervinus, sample 21 and 22, which are Pluteus petastasis, they are equally similar. They all have a % similarity of 97.47% similarity to Mike's sample. So according to the % similarity, these are all equal candidates as the best matches. And this indicates that Mike's sample is probably in the genus Pluteus and maybe is one of those two species, but we can't be confident which of the two it is from this alignment. Now that you've aligned and compared your sequences in the muscle alignment, you can begin to build phylogenetic trees from those sequences. Phylogenetic trees are a graphical representation of relationships among taxonomic groups. DNA Subway uses a tree building algorithm called PHILIP, or phylogenetic inference package, to attempt to reconstruct the order in which sequence mutations accumulated as different lineages diverged from a common ancestor. This software will create neighbor joining, or NJ, and maximum likelihood, or ML trees, by analyzing the similarities and differences among the DNA sequences you ask it to compare. These two types of trees are constructed using different methods, with NJ trees tending to be a bit simpler to interpret at the cost of being a little less accurate relative to the ML trees. There's two major things that you'll need to provide the software if you want to create a phylogenetic tree. Number one is you'll need to input known barcodes of closely related species. In other words, your blast hits. And then you'll also need to have known barcodes of a more distantly related species for comparison or rooting the tree. That's called the outgroup. This helps root your tree by determining which nucleotide changes are more ancestral versus derived. Let's make an ML tree first. The ML tree will automatically analyze all the taxa that were present in the muscle alignment you previously constructed. All you have to do is select an outgroup to root the tree. In this case, we've selected smut fungus. How did we know to use the smut fungus as the outgroup? Hit pause until you know the answer. If you said because it was the least similar to all the other taxonomic groups that we looked at in our percent similarity matrix, you are correct. This is the maximum likelihood tree output. This algorithm fits mutations to a tree to produce the most parsimonious tree. In other words, the one that requires the fewest number of mutations to explain the sequence data, aka the shortest branch lengths. Internal nodes on this tree, represented by the big blue dots, represent a common ancestor shared by taxa on the branch tips. Horizontal lines represent sequence differences, aka evolutionary change. The longer the horizontal branch is, the larger the evolutionary distance, aka the less related those taxa are. For example, you can see that Mike's sample has a very short branch off the Pluteus cervinus and Petacetus clade, and therefore is closely related to those two species. In contrast, the smut fungus down at the bottom, that outgroup has a very long branch, as the outgroup should, demonstrating its distant relation to all the other taxa. According to this tree, which species is most closely related to trays unknown? Hit pause until you know the answer. If you said the genus Hymenopellis, you are correct. We can't say for sure which species it is because tray is about equally related to the Limnospora, Megalospora, Incognita radica, and Orientalis. How about Mike's? Which species is Mike's sample most closely related to? Go and hit pause until you know the answer. If you said the genus Pluteus, you would be correct. We can't say for sure whether it's more closely related to Cervinus or Petacetus, but it's got to be one of those two, according to this tree. How about Jessica's sample? Which species is most closely related to hers? Hit pause until you know the answer. All right. If you said Pluteus ongoi, you would be correct. She is very closely related to just that species. Everything in that clade is just that single species. And how about Luisa's sample? Go ahead and hit pause until you know the answer. If you said Grifola frondosa, you would be correct because that is the only species in that clade and she is very closely related to that clade. Next, let's make an NJ tree. Same as the ML, the software will automatically analyze all the taxa that were present in the muscle alignment you previously constructed. All you have to do is select an outgroup to root the tree and hit select. We'll use the same outgroup as before, as the smut fungus is still the least similar taxon from the percent similarity matrix in the muscle alignment. Your output should look something like this. The neighbor joining algorithm builds a tree from the bottom up by comparing the evolutionary distance between pairs of DNA sequences. Sequences with best matching sequences are linked as neighbors that share common nodes in the tree. Because the branch distances are produced in a pairwise manner, neighbor joining does not optimize branch length and tree parsimony. The numbers you see on the tree typically represent bootstrap values. These values indicate the confidence level in a particular branch of the tree, reflecting how frequently percentage-wise that branch appears in multiple trees generated from resampling your dataset over and over again. Higher bootstrap values suggest a more reliable branch. For example, a bootstrap value of 95 means that 95% of the resampled trees contained that specific branch in that specific location, indicating a high level of confidence in that branch's existence in the quote-unquote true tree, if such a thing exists. A common rule of thumb is that values above 70 or 80% in bootstrap are generally considered well supported, though this can vary depending on the specific study and data set that you're looking at. So according to this neighbor joining tree, which species is most closely related to Mike's unknown? Go ahead and hit pause until you know the answer. If you said the genus Pluteus, you'd be correct. We can't say the species for sure. And that's because if you look at Mike's sample, it's sort of an outgroup to the Pluteus pedicetus and Pluteus cervinus clade, and it's equally related to each of those different species. In addition, the next closest related sort of sister clade is the one with Jessica's sample in it that has all Pluteus ongoi. And so once again, it's a member of the genus Pluteus, just a different species. So Mike's sample is almost certainly a member of the genus Pluteus, but we can't say for sure which species it is. Next, let's look at Trey's sample. According to this tree, which species is most closely related to Trey's unknown? Go ahead and pause until you think you know the answer. All right, if you said the genus Hymenopellis, you would be correct. We can't say for sure which species Trey's sample is, but we can say that it's very closely related to different species, all members of the genus Hymenopellis, whether it's Orientalis, Radicata, Limnospora, or Megalospora, or Incognita. Trey is certainly within a clade that's only has members of that genus, and almost certainly a member of that genus as well. How about Jessica's sample? According to this tree, which species is most closely related to Jessica's unknown? Go ahead and hit pause until you think you know the answer. All right, if you said Pluteus ongoi, you would be correct. Jessica's sample is more closely related to Pluteus ongoi samples than another sample of the same species even is. And so because she's sort of contained within this clade that only has members of that species and no other species, we can be pretty confident that her sample is the same species. How about Louisa's sample? So Louisa's sample is down towards the bottom. Go ahead and pause until you know the answer. All right, if you said Gryphula frondosa, you would be correct. Same story as Jessica's sample. Everything in that clade with Louisa's sample is in the same species, Gryphula frondosa, and in fact, Louisa's sample is more closely related to a Gryphula frondosa sequence than another known sequence of the same species, or two other known sequences of the same species are. So we can feel pretty confident that Louisa's sample probably is that species, Gryphula frondosa. All right, so how do we put all of these pieces together to ID our unknown samples? Let's start with Mike's sample. Here's the photo he took from the sample they used to create his DNA barcode. And let's look at the evidence we have from our DNA barcoding analyses. Number one, we have our best blast hits. According to the percent sequence similarity matrix, Pluteus cervinus and P. pedicetus were our two best blast hits. The alignment lengths were 593 base pairs, which beat our 400 base pair cutoff to be labeled as quote-unquote trustworthy. And when we looked at our number of mismatches divided by our alignment length for those blast hits, they were about 13 mismatches for those 593 aligned base pairs, which is about 2% mismatch, which is greater than our 1% cutoff to ID to the species level. When we look at our overall percent sequence similarity, Pluteus cervinus and P. pedicetus both had a 97.96% similarity, which is, of course, very good, but also less than our 99% rule of thumb needed to ID to the species level, or typically used to ID to the species level. When we look at the results of our NJ and ML trees, both P. cervinus and pedicetus were the closest relatives to Mike's samples. They were equally related in both the NJ tree and ML tree analysis. Furthermore, if you go and Google search images of those two species, cervinus and pedicetus, this is what the images look like. And they look very similar to Mike's sample. So it's difficult morphologically speaking, without having a sort of more expert level of knowledge. It's difficult to tell which of those two it might be. So that doesn't help us discriminate further. Likewise, if we look at the known geographic ranges of P. cervinus and Petacetus, they're both all over the eastern North America, so they're within the range of where we would have found them. So that doesn't help us eliminate which one of these species it might be either. So in summary, our BLAST, percent similarity, neighbor joining, and ML trees all give sort of similar info in the sense that it's going to be one of those two species, but we can't be sure which. So we can only be sure that Mike's sample is in the Pluteus genus. Photos and range, geographic ranges, also support this idea that it's in the Pluteus genus, and it's likely one of these two species, either Cervinus or Petacetus, but we can't say which for sure from the data that we have up to this point. Next, let's look at Jessica's sample and the evidence we have from our DNA subway analyses. Her best BLAST hits were Pluteus ongoi. The alignment length was 588 base pairs, which beats our 400 base pair cutoff to be trustworthy. There were 0 mismatches in this alignment, so there's a 0% difference between that and the BLAST hits, so it's definitely below our cutoff to ID to the species level. Percent sequence similarity was 100% similar to 2 different Pluteus ongoi samples, so once again, this beats our 1% cutoff to ID to the species level. Our NJ tree and ML tree both support Pluteus ongoi as the closest relative and IDing it to the species level. When you do a Google or INAT search for images of that species, you can see that morphologically speaking, it's very similar to the photo that Jessica took, so that also supports it being this species. And if you look at the range map of Pluteus ongoi, they're found all over eastern North America, even here in the Harrisonburg area. So all of these pieces of information, using the totality of information together, that all supports her sample being Pluteus ongoi. Our BLAST, percent sequence similarity, neighbor joining tree, ML tree, all support IDing the sample to Pluteus ongoi. Sequence length, sequence quality, the number of mismatches, the percent similarity all exceed the rough thresholds to ID to the species level, and our photo and range maps match. So the conclusion we can probably safely draw from this is that Jessica's sample is likely the species Pluteus ongoi. So the challenge I pose to you is to collect all of that information to ID Louisa's sample. And I am not going to provide you the answers. It's up to you to figure that stuff out and draw your own conclusions. And then do the same for Trey's sample. And once again, I'm not going to provide the answers in this case. It's up to you to figure it out and do your best. Good luck.